Welcome to Matri Academy. Let's begin the chapter 24, Nitrogen Fixation. The synthetic process of reduction of atmospheric nitrogen to ammonia is called nitrogen fixation. The crop associated with nitrogen fixation is legume crops. Question asked in ICAR JRF 2003. Nitrogen fixing prokaryotes are called as diazotrophs. Most of nitrogen is absorbed by plants in the form of nitrate. ARS 2007 question. Nitrification means conversion of ammonia into nitrate. IARI 2003 question. Acidic medium is the best for nitrate uptake. Question asked in ICAR JRF 2004. Nitrate reductase requires molybdenum and nitrite reductase requires flavin FAD. Free living nitrogen fixing bacteria. Anaerobic. Clostridium species, Azoospermum species, Bacillus polymixa, Klebsiella pneumoniae. And aerobic species are Azotobacter species. Azotobacter is a free living aerobic nitrogen fixing bacteria. Question asked in ARS 2007. Azotobacter is an aerobic free living nitrogen fixing bacteria, while Clostridium is an anaerobic free living nitrogen fixing bacteria. Question asked in IARI 2000 and 2004. Frankia is a symbiotic nitrogen fixer, while Klebsiella is a free living nitrogen fixing bacteria. Question asked in IARI 2003. Rhizobium is involved in symbiotic nitrogen fixation. ICAR JRF 1995 question. Rhizobium is a gram-negative motile rod-shaped proteobacteria. Associative nitrogen fixer is Azoospermillium, while Azotobacter is a free-living aerobic and Clostridium is free-living anaerobic microorganisms. Question asked several times in ICAR JRF 2000, IARI 2001. In most nitrogen-fixing microorganisms, reduced ferredoxin acts as a donor for electrons. One molecule of nitrogen fixation requires 16 ATP molecules, as you can see here in the reaction. 8 electrons are required to convert one molecule of nitrogen into 2 molecules of ammonia. Ferrous molybdenum is found in enzyme nitrogenase. Question asked in ARS 2007. Molybdenum-rich soils have less accumulated nitrogen as it helps in nitrate reduction through nitrate reductase enzyme. Molybdenum-deficient soil plants accumulate nitrogen. Molybdenum and calcium helps in nitrogen uptake. IARI 2000 question. Nitrogen fixation is enhanced by the uptake of molybdenum by the plants. ICAR JRF 2001 question. The enzyme nitrogenase, which is involved in nitrogen fixation, is a heteromeric dimers. ARS Net 2001. Nitrogenase is having a ferrous molybdenum complex. ARS Net 2001. Heme of leg hemoglobin is synthesized by rhizobium. Leg hemoglobin is synthesized in nitrogen fixing nodule. ARS Net 2001 question. Leg hemoglobin is synthesized by plants in response to rhizobium infection symbiotically. The pigment involved in nitrogen fixation in leguminous plant is leg hemoglobin. Leg hemoglobin is present in nodulating bacteria to prevent the reaction of oxygen with the enzyme nitrogenase reductase. ARS Net 2001 question. Leguminous plants maintain a very low concentration of free oxygen in their root nodule by binding oxygen to leg hemoglobin, a pink color compound, a homologue of hemoglobin which is red color compound. The nitrogen fixing enzyme nitrogenase reductase is sensitive to oxygen, so leg hemoglobin is there in root nodules to combat the negative effect of oxygen on the activity of enzyme. Question asked in ICAR JRF 2004. Symbiotic association is there between rhizobium and legumes. ICAR JRF 2001. Root exudates induce the nodulation gene responsible for nodule formation. These NOD genes synthesize nodulation factor or NOD factors. In the infected cells, the rhizobia divide and enlarge to form bacteroids. Reduction of nitrogen to ammonia occurs in bacteroids. Only one leguminous genus, Parasporia, is known to form root nodules with a rhizobia symbiont. Bacterial genes concerning symbiosis are collectively referred to as SYM genes. During symbiotic nitrogen fixation process, calcium plays a crucial role in modifying the ability of root to absorb rhizobia. The rhizobia bacterial cells enlarge in size inside the nodules and assume pleomorphic shapes and are called as bacteroids. Question asked in IARI 2003. For the reduction of one molecule of nitrogen into two molecules of ammonia, it requires 12 molecules of ATP during symbiotic nitrogen fixation and 16 molecules of ATP during asymbiotic nitrogen fixation. Ammonia is converted into nitrite by nitrosomonas and nitrosobacter, and then nitrite is converted into nitrate by nitrobacter. Question asked various times in ICR JRF 2001, 2003, IARI 2003 and ARS 2018. The slowest growing species of rhizobium is Rhizobium japonicum, the cow pea rhizobium, now also known as bradyrhizobium. Cyanobacteria produce nitrogenase and nitrogen fixation occurs in specialized structures are called heterocyst. Biological nitrogen fixation is also called diazotrophy. Nitrogen fixing capacity of blue-green algae can be enhanced by the application of phosphorus and molybdenum. Nitrogen fixation interaction between bacteria and leguminous plants is due to NIF genes. NIF genes are associated with rhizobium bacteroid. NIF genes are involved in the synthesis of nitrogenase and the catalytic process of nitrogen fixation. Question asked in ARS 2013. Ammonia is excreted as ammonia in aminotelic organism, urea in ureotelic organism, and uric acid in uricotelic organism. Molecular biology of nitrogen fixation. These nitrogen fixation are well studied in Klebsiella pneumonia NIF genes. Merrick identified 14 NIF genes in Klebsiella pneumonia which were organized into 8 operons. NIF genes are located on megaplasmid. 14 genes involved organized into 8 transcriptional units in NIF clusters. KDH gene, enzyme complex of 2 components. Nitrogenase is a tetramer consisting of 2 units of ferrous molybdenum and 2 units of ferrous proteins. And nitrogenase reductase contains 2 units of 1 polypeptide. Enzyme nitrogenase reductase is a homomeric dimer of identical 30 kilodalton subunits bridged by 4 ferrous sulfur clusters. NIF-K codes for 1 subunit of nitrogenase, NIF-D codes for 1 subunit of nitrogenase, NIF-H code for component of nitrogenase reductase, and NIF-M component which makes nitrogenase reductase an active enzyme. NIF-E codes for synthesis and addition of the ferrous molybdenum protein to the pro-nitrogenase molecule. NIF-A and NIF-L involved in regulation expression of all the NIF gene products. NIF gene cluster is located on large megaplasmid of rhizobium. Question asked in ICR JRF 1999. Regulator of NIF gene cluster is NIF-A and L, ICR JRF 99. Accessory genes are called fixed genes. They are also necessary for the function and regulation of nitrogenase in anaerobic and aerobic nitrogen fixing bacteria. Nitrate is the most inhibitory to nodulation. ICR JRF 2004 question. Nitrate reductase cofactor is molybdenum. ICR JRF 99 question. Nitrate reductase uses NADH as electron donor. Nitrate reductase is found in cytosol of plant cells. ICR JRF 2003 question. Elements which is actively involved in nitrate reduction and nitrogen fixation is molybdenum. ICR JRF 2000 question. First amino acid produced during nitrogen fixation is glutamine. ICAR JRF 99 question. First organic compound which is formed after ammonia assimilation is glutamine. ICR JRF 2004 question. Nodulin genes are found in plants. ARS Net 2001 question. Under excessive nitrogen supply, the enzyme glutamate dehydrogenase plays an important role. The energy for the enzymatic split of nitrogen comes from ATP. ICR JRF 2004 question. Bacteria responsible for nitrogen fixation having purple color and can also photosynthesis is reduced pseudomonas species. Nitrogen fixation in rice field occurs due to the presence of Anabena blue-green algae. Number of ATPs required for sulfur assimilation is 14. So these all are the short notes about the nitrogen fixation bacteria and about the molecular biology of nitrogen fixation. So let's tune to my channel. Always like and subscribe.